Hello everybody, this video will be about adaptive immunity. We will talk about antigens, antigen-presenting cell, major histocompatibility complex, and T lymphocytes. Okay, as you know, in our body we have two types of immune systems, the innate immune system and the adaptive immune system. Innate immune system is an antigen non-specific, and it's a kind of immediately immune response which start to work very quickly within several hours after exposure to almost any microbe, and it's a primary immune system. Innate immunity is the immunity one is born with, and is the initial response by the body to eliminate microbes and prevent infection. So preventing this kind of immunity start to prevent infection, and it's the first line of the protection in our body. But adaptive immunity is antigen specific defense. It means that to develop adaptive immunity, it is necessary to recognize the antigens, and adaptive immunity is developed against specific antigens, and it can interact with one antigen, and this kind of immunity takes several days to become protective, so it is not immediately, and work of adaptive immunity is to remove specific antigen. This is the immunity one develops through our life. It's experience of it's an individual experience. There are two major branches of the adaptive immunity response: humoral immunity and cell-mediated immunity. Humoral immunity is associated with antibody production, and antibody production is a response to antigen, and it's by B lymphocytes, and cell-mediated immunity associated with production of cytotoxic T lymphocytes or T killers, and in this type of immunity, also macrophage, natural killers, and cytokines and T lymphocytes take part. Okay, but as you understand now, antigen, it's a main particle, it's a main participant in the adaptive immune response. So antigen stimulate adaptive immune response to start to work. What is antigen? Antigen is any substance, usually foreign, with the ability to stimulate an immune response when presented in an effective fashion. And antigen are activators of the adaptive immune defenses. Adaptive immune defense always starts with the antigen recognition. The term antigen comes from a combination of the word antibody and generator, and the molecule that simulates antibody production is said to be antigenic. However, antigen also play an essential role in stimulating cellular immunity, so to stimulate production of T cytotoxic lymphocyte, T killer. And for this reason, antigens are sometimes more accurately referred to as immunogens. Not all molecules are immunogens. In general, antigens are proteins because proteins are best immunogens than carbohydrates, Um, and also, heptans are incomplete immunogens because they are often too small to immunize. So they have a very small molecular weight, and that's why they can't induce the adaptive immune response. But if they connect with something like adjuvants, they can become to be immunogenic. Uh, so heptans are also antigens, but not complete. So they need to attach to the carrier molecule, such as a protein. Okay, like that. This is a heptan, and heptan have a so little molecular weight. That's why it can induce immune response, and it needs carrier molecule. And when it attached to the carrier molecule, it became to be immunogenic and can induce immune response, adaptive immune response. Okay, so to be immunogenic, an antigen must possess three characteristics: to be of high molecular weight, exhibit chemical complexity, exhibit foreignness. Uh, it is necessary to be recognized as a non-self by the body. Each antigen contains actual antigenic determinant or epitope. An epitope, it's an antigenic determinant. It's a part of the antigen which is in contact with antibody, which induce the immune response. And epitope, it's a specific part of antigen. Um, and it is a actual molecular structure that interacts with a single antibody molecular antibody like T cell receptor. Any given antigen may have several epitopes. Each epitope is recognized by a different antibody or TCR. Okay, look at this picture. Here we have an antigen, and this is a carrier part, and this is the epitopes. You can see that epitopes can be not only one, but two or three or more. And look here, antibodies, they interact with the epitope, not with the carrier part of the antigen, but with epitope usually, always. And also on this picture, you can see bacterial cell. And this bacterial cell have different antigens. And these antigens have a epitopes, and antibody also interact with epitopes. Okay. Antigen classification. So the main classification is by the foreignness. So endogenous and exogenous. Endogenous antigens are protein found within the system of the human cell. Exogenous antigens are antigens that enter from outside the body, such as bacterial, fungal, viral, protozoan, and other. This exogenous antigen enter macrophage, dendritic cell, and B lymphocytes through phagocytosis or pinocytosis. Outer antigens are any of an organism's own antigens, like self-antigens, that stimulate an autoimmune reaction that is humoral immunity or cell-mediated against self. Example of endogenous antigen include viral proteins produced during viral replication, proteins produced by intracellular bacteria such as Rickettsia or Chlamydia during their replication. Proteins that have escaped into the cytosol from the phagosome or phagocytes, such as antigen-presenting cell, tumor antigens, and self-peptides from the host cellular proteins. Exogenous antigen are bacterial, fungi, protozoan, and free viruses. These exogenous antigens are enter to the macrophage, dendritic cell, and B lymphocytes through phagocytosis. The microbes are engulfed in and placed in a phagosome, which then fused with lysosome. Following this fusion, the phagolysosome becomes acidified. So I just described the process of phagocytosis. Acidification in turn activates the protease within the phagolysosome, enabling protein antigens from the microbe to be degraded in a series of short peptides. So it's a process of destroying of bacteria and taking its epitopes, its antigens. These peptides epitopes are then attached to the MHC second molecules and are then transported to the surface of the antigen-presenting cell, APC cell. So any exogenous antigen must be presented by the antigen-presenting cell to induce immune response, adaptive immune response. And when antigen-presenting cells catches a new pathogen, it is necessary to destroy it, to take it antigen, and to put its epitope of the new pathogen to the antigen-presenting receptor, MHC second receptor. Autoantigens are tissue which are strictly isolated from the host immune cells, such as thyroid gland, crystalline brain tissue, testicles, and any damage of enumeration tissue leads to production of autoantibodies and development of autoimmune disease. Okay, bacterial antigen. Everything in bacteria can be antigenic, like capsule, like flagella, some substance from cell wall, O antigen, decoy acid, and some secreted antigen like toxins and enzymes. Okay, here you can see O antigen, it's an antigen of cell wall. H antigen, it's an antigen of flagella. K antigen, it's an antigen of the capsule, and V antigen in case of salmonella, it's a capsule antigen. Okay, look at this picture here. You can see the cell wall of gram-negative bacteria, and if you remember, gram-negative bacteria, they have thin cell wall, and in their cell walls there is outer membrane, and this outer membrane consists of lipopolysaccharide, and lipopolysaccharide have three parts. Lipid A, which is a endotoxin, and the core part, just to connect lipopolysaccharide, and polysaccharidic part, it's a O antigen. This is O antigen, main antigen of all gram-negative bacteria. It is on the surface of the gram-negative bacteria cell wall. Okay. Okay, and also you should know that some bacteria can can synthesize superantigen. Superantigen is a class of antigens that induce non-specific activation of T cell, resulting in polyclonal T cell activation and excessive uncoordinated cytokine production. And also protective antigen. This is the main antigen of the pathogen, and if this antigen will be neutralized, infection will not be occur. So protective antigen is a substance prepared from pathogen which, when injected into the host, induce the production of the antibody, enough for production of antibody for protection for the given pathogen. So when scientists create a new vaccine, they need to find protective antigen in the pathogen and then put it to the vaccine. So any vaccine must contain protective antigen because protection in the human body will be occurs against only protective antigen. And also iso antigen or allo antigen, an immunological active protein or polysaccharide present in some but not all individuals in a particular species. This substance initiates the formation of antibody when introduced into the individuals of the species that genetically lack to the isoantigen. Like all antigens, they are also active in stimulation antibody production in heterological species. Example, the APO and air blood factors histocompatibility antigen. Isoantigen vary in their potential to cause transfusion reaction when mismatched blood is given. Okay, next question is major histocompatibility complex. The major histocompatibility complex MHC is a collection of genes coding for MHC molecules found on the surface of all nucleated cells of the body. So all nucleated cells of the body contain these MHC molecules. In human, the MHC genes are also referred to as human leukocyte antigen, HLA genes. Mature red blood cells, which lack a nucleus, are the only cells that do not express MHC molecules on their surface. You should remember, you should notice that all cells in human body contain MHC on their surface. One except one cell, erythrocytes. MHC molecules enable T lymphocytes to recognize epitope in this, and discriminate self from non-self. Okay, so there are two classes of MHC, MHC1 and MHC2. MHC1, the molecules are found on all nucleated cells. They present normal self-antigen as well as abnormal or non-self pathogen to the effector T cell involved in the cellular immunity. MHC1 molecules present epitopes to T8 lymphocytes. So with these molecules, human cell will show that it is self cell, or also it can show with this molecule that this cell is infected or became tumorous. And these molecules connect with the T cell, T8 cell, T killer. And also natural killer, they also check MHC1. So two killer cell, natural killer and T killer, they check MHC1 with one difference. Natural killer, they just check, is it normal or abnormal? Normal means this is normal cell. Abnormal means that this cell is infected with viruses, for example. Or abnormal MHC1 means that this cell became tumorous, became abnormal. And if natural killer find abnormal MHC1, it will destroy the cell. But T killer, they are specific cell. So they are part of adaptive immunity. That's why T killers they check MHC1, but they need to find specific antigen. If they will find specific antigen in pathogen antigen of the pathogen in the MHC1 of the cell, they will recognize this cell as a infected and will kill. Okay, MHC second, they are different from the MHC1 because they are present on the surface of all antigen presenting cell. Antigen presenting cell, and by this molecule, antigen presenting cell will show a new antigen to the T helper. So MHC second molecules present epitope to T4 lymphocyte T helper. So, the expression of MHC molecules is increased by cytokines produced during both innate immune response and adaptive immune response. So there are in the human body some cytokines, some molecules by which immune cell interact with each other. So they use cytokines as a messenger. If one immune cell need to send the message to us to other immune cell, it will release the cytokine and send it to other cell, and other cell will catch with specific receptor this cytokine and will understand the signal. Cytokines such as interferon alpha, interferon beta, interferon gamma, tumor necrosis factor increase the expression of MHC1 molecules, while interferon gamma is the main cytokine to increase the expression of MHC2 molecules. Both types of MHC molecules are transmembrane glycoproteins that assemble as dimers in the cytoplasmic membrane of cell, but their structure are quite different. Okay, look at this picture. Here are MHC1 and MHC2. MHC1 presence on the surface of all nucleated cells except erythrocytes. And with this receptor, human cell will show that it is normal, it is non-infected, it's a self-cell. Natural killer and T killer check this receptor to understand that it is self or not self, normal or abnormal. And class second, it's a receptor which will present on the surface of antigen presenting cell. And with these molecules, antigen presenting cell show a new antigen with its epitopes to T helper to induce adaptive immunity. Okay. MHC1 molecules are designed to enable the body to recognize infected cell and tumor cell and destroy them with cytotoxic T lymphocyte T killer or CTLs. CTLs, T killer, are effector defense cells derived from naive T8 lymphocytes, bind peptide epitopes typically from endogenous antigen. MHC second molecules are designed to enable T4 lymphocytes to recognize epitopes of exogenous antigen and discriminate cell from non-cell. MHC second molecules are made by antigen presenting cell or APC such as dendritic cell, macrophage, and B lymphocytes. Present MHC second peptide complex to naive T4 lymphocytes or effector T4 lymphocytes that have a complementary shape T cell receptor or TCR, being peptide epitopes typically from exogenous antigen. Okay, you can see the process of process of antigen processing of the antigen and binding of peptide epitopes from endogenous antigen to MHC1 molecules by dendritic cell endogenous to antigen, and those located within cytosol to the cell to the body. Okay. So this is a process of antigen preparation. So this is a antigen presenting cell. It catch a new pathogen, new antigen, pathogen which is never met before, have never met before. Okay, so it catch it and make a phagosome and destroy it, and take its main antigens, its epitope, and also antigen presented cell create a MHC receptor, MHC molecules, and put epitopes of new antigen to the MHC second molecules, and then put MHC second with the new epitopes on its surface. So antigen presented cell will present a new antigen to the adaptive immune system cell. And also, as I said previously, all cell all cell um, all cells that have a nucleus except erythrocyte have a MHC1 class. So antigen presenting cell will show the antigen in MHC second receptor, and also it will show this antigen in the MHC1. I will explain later why it is so important. Okay, antigen presenting cell or nucleated cell in body have a mechanism for processing and presenting antigen in association with MHC molecules. This signals the immune system indicates whether the cell is normal and healthy or infected within an intracellular pathogen. However, only macrophage, dendritic cell, and B cell have the ability to present antigens specifically for a purpose to activating T cell, T helper, I mean, to create the adaptive immune system. So let's discuss each antigen presenting cell. Dendritic cell are derived from monocytes and are referred to as a myeloid dendritic cell. Dendritic cell function as antigen presenting cell. Dendritic cells are present in small quantities in tissues that are in contact with the external environment, mainly in the skin, where there is a specialized dendritic cell type called Langerhans cell, and the inner lining of the nose, lungs, stomach, and the intestines. So they are on the first line, and they are the first who met the new antigen. Once activated, the dendritic cell migrate to the lymphoid tissue where they interact with T cell and B cell to initiate the adaptive immune response. So after interaction with the new antigen, they will go to the lymphoid system to initiate the adaptive immune response. So look at this. Look at this picture. Here you can see the antigen presented cell. It catches the pathogen, then it put this pathogen to the phagosome. So the phagocytosis occur, then the process of phagosome and lysosome fusion, and destroying the bacteria. But look here, bacterial antigens are kept safely, so they stay as it was before destroying. So destroy only pathogen, but keep its antigen. And then antigen presented cell put this antigen to the MHC second molecules and put this complex on the surface of the on the surface of the antigen presenting cell. This is very important. Also, macrophage cell produced by the differentiation of monocytes in tissue also. So dendritic cell and macrophage, they both belong to the monocytes. These specialized phagocytic cells that ingest microbes and particulate antigens digest them in phagolysosome present fragments in the cell surface on MHC second molecules. So they work, macrophage and dendritic cells, they do the same work, the same job. But lymphocytes, they are different than macrophage and dendritic cell. Like all lymphocytes, B lymphocytes circulate back and forth between the blood and the lymphoid system of the body. B lymphocytes are able to capture and present peptide epitopes from exogenous antigen to the effector T lymphocytes. The MHC second molecules bind peptides epitope from exogenous antigen and place them on the surface of the lymphocytes B. Here, the MHC second peptide complex can be recognized by completely shaped T cell receptor TCR and CD4 molecules and effector T4 lymphocytes. This interaction eventually triggers the effector T lymphocyte to produce and secrete various cytokines that enable that B lymphocyte to proliferate and differentiate into the antibody secreting plasma cell. B cell can bind to a specific antigen through the surface of immunoglobulin A or immunoglobulin G. Internalize it, degrade it into peptides which associate with MHC class second molecules. So look at these three types of antigen presented cell. This is macrophage, and this is dendritic cell. So macrophage, they catch some pathogen, it puts them to destroy them, and then put its antigen to the MHC second class receptor, and then show new antigen to the T helper. Dendritic cell, they also do like macrophage. They catch some pathogen and then put in MHC second receptor and then show to the T helper. But B cell, they are not able to engulf something. They are not able to work as a phagocytic cell. So they not eat a pathogen, but it can catch pathogen by its BCR receptor. BCR receptor, it's a receptor of the B cell. And after catching antigen, it take it to the inside the B cell, inside the cell, destroy two peptides and put to the MHC second receptor and interact with T helper, activate T helper. Antigen processing. Antigen processing involves degrading the antigen into peptide fragment. The vast majority of epitopes recognized by T cell are fragments from a peptide chain. Only a minority of peptide fragments from a protein's antigen are able to bind to a particular MHC molecule. Okay. Lymphocytes. Let's talk about lymphocytes clonal selection. Lymphocytes, T and B cell, specifically recognize foreign material or antigen. B cells are responsible for the production of antibodies that act against extracellular pathogens. T cells are mainly concerned with the cellular immune response to intracellular pathogens like viruses. They also regulate the responses of B cell and the overall and the immune response. Tolerant cell, T and B cell are derived from bone marrow stem system, but T cell then develop in the thymus, while B cell develop in the bone marrow. B bone marrow, T thymus. Both T and B cell have a tolerance select. So two kinds of selection. Central tolerance is the mechanism by which newly development T cell and B cell are rendered non-reactive to non-cell tools. Okay, and central tolerance occurs while cells are still present in the primary lymphoid organ, thymus or in the bone marrow. Clonal selection occurs after immature lymphocytes express antigen receptor. T cell with useful receptors are preserved, and mainly potential harmful self-antigen reactive cells are eliminated by the process of selection induced by antigen receptor engagement. So the main significance of the selection in the central in the primary immune organs is destroying of self or outer reacting outer reacting lymphocytes to destroy, to kill the all cell which are outer reactive, which can react with the outer antigen. The preservation of useful specificities is called positive selection. Positive selection ensures maturation of T cell whose receptor bind weakly to self-major histocompatibility complex molecules. Negative selection is a process that eliminates developing lymphocytes whose antigen receptor binds strong to self-antigen present in the lymphoid organs. Peripheral tolerance is generated after the cell reaches the periphery. Tolerance is needed to protect against autoimmune disease. So here we can see the process of selection, clonal selection, and clonal selection of the B and T lymphocytes. The first hematopoietic stem cell, the hematopoietic cell from which all blood cell will be created. And second, it's a immature lymphocyte with various receptors. And here you can see self-antigen. These red particles are self-antigen from the body's tissue. And here we can see the lymphocytes which can catch and recognize, which can react with the self-antigen. They need to be destroyed because they will be autoreactive T lymphocytes. And then you can see a lot of T cell will be destroyed in the process of clonal selection. Okay, receptors. So T cells, they have T cell receptor TCR, and is involved in the first step of pathogen epitope recognition during the activation process. Activation and differentiation of helper T cell. So T cell, they can be T helper, CD4 cell, or T killer, CD8 cell. So T helper, it's a cell which interact with APC, antigen presented cell. And they are in the first line of the inducing of the adaptive immune process, adaptive immune response, because they interact with antigen-presented cell, and they recognize new antigen in the MHC second receptor of the APC cell. The first step in the activation process is TCR recognition of specific foreign epitope presented with the MHC second antigen binding cell cleft. The second step involves the interaction of CD4 and the helper T cell with the region of the MHC second molecules separates from the antigen binding cleft. This second interaction encodes the MHC second TCR complex and ensures that the helper T cell is recognized as both the foreign non-self epitope and self antigen of the APC. Third step, the APC and T cells secrete cytokines that activate the helper. The activated helper T cells then proliferates, dividing by mitosis to produce clonal naive helper T cells that differentiate into subtypes with different function. Look here. This is antigen-presented cell. It catch some pathogen, some new pathogen. It destroyed and put its epitope to the MHC second molecules. And then it goes to find T helper. When it finds T helper, it show a new pathogen by MHC second molecules. T helper have a T cell receptor. T cell receptor interact with MHC second molecules and recognize a new antigen, new epitope. And then naive T helper activated and start to proliferate to another kind of T cell, to be T helper 1, T helper 2, and memory T helper cell. So it proliferated into three kinds of the cell. T helper 1, which will take part in the cellular adaptive immune answer. T helper 2, which will take part in the humoral adaptive immune answer, and the T helper memory cell, which will take part in the secondary immune response. Okay, so this is a step of initiation of adaptive immune system. Any adaptive immune response, any specific immune response, start with this process. The process of recognition of T helper, of new pathogen from by interaction with the antigen presenting cell and its MHC second receptor. Okay. Here you can see that T helper can be like T helper 1, T helper 2, T helper 17, and memory T helper. So T helper 1 take part in the stimulate the activation of the T killer, T cytotoxic cell, and so it initiate the process of cellular specific immune answer development. T helper 2, they stimulate B cells to be activated and to produce antibodies, so they take part in the humoral specific immune answer. And T17, they stimulate immune to specific infections such as a chronic mononucleation infection. And memory T helper, they remember a specific pathogen and mount a strong rapid secondary response upon re-exposure. Activation and differentiation of cytotoxic T cell. Cytotoxic T cells, also referred to as cytotoxic T lymphocytes or CTLs, are activated by APC in a three-step process similar to that of T cell. The key difference is that activation of cytotoxic T cell involves recognition of antigen presented with the MHC1 as the opposite of MHC2 and interaction of CD8 as the opposite to CD4 with the receptor complex. After the successful co-recognition of foreign epitopes, the self-antigen, the production of cytokines by APC and cytotoxic T cell activate clonal proliferation and differentiation. Activated cytotoxic T cell can be differentiated into effector cytotoxic T cell that target pathogen for destruction or memory cells that are ready to respond to subsequent exposure. As noted, proliferation and differentiation of cytotoxic T cell is also stimulated by cytokines secreted from T helper 1 cell activated by the same foreign epitope. So after activation, the dendritic cell produce cytokines that contribute to proliferation of T8 lymphocyte and their differentiation into effector T4 lymphocyte called cytotoxic T lymphocyte (CTLs) that are able to bind and kill infected cells, and tumor cells display the same peptides MHC1 complex on their surface. Once an infected cell is recognized, the TCR of the cytotoxic T cell binds to the epitope and release perforin and enzymes that destroy the infected cell. Some of the T8 lymphocytes differentiate into circulated T8 memory cells that enable a more rapid and greater production of CTLs upon subsequent exposure to the same antigen, so in the secondary immune answer they take part. Okay, and I will explain you here. Okay, so look at this picture. Uh, So, activated T cells, they need to recognize MHC1 receptor. That's why they need to interact with the antigen presenting cell with its MHC1 receptor. MHC1 receptor, but before that, T helper must be activated with uh, with the antigen presenting cell. So antigen presenting cell, they activate T helper, then T helper, like I showed here previously. I will show here. Oh, okay. Uh, so after initiation of this process, T helper, they proliferate to the T helper 1, and T helper 1, they send a signal to the, oh, okay, they send a signal to the CTL cell. So the process of activation of the CTL of the T killer, it start with the activation of the T helper. After activation of the T helper send a signal to the cytotoxic lymphocyte T killer, and they start to recognize MHC1 receptor of the antigen presenting cell. You know that MHC1 receptor is present on each cell except erythrocytes. That's why CTL, they interact with the antigen presented cell, but not with the MHC second, but with MHC1. Recognize, study, research new epitope, and then became to be activated. Activated CTL start to proliferate to make new clones. All that cell will be like army, like warriors, and all the cell will have information about new pathogen and its epitope. And all these clones will go into the blood and will start to find infected cell. They will check each cell with its MHC1 receptor. And if they will find this peptide in the MHC1 receptor on the surface of the cell, they will recognize the cell as infected and they will destroy it by enzymes and perforins. Okay. Next, in the second part of the lecture, we will talk about B cell activation. Adaptive immunity will be in two parts in two video.